All right, class. So welcome to week 5. You guys are doing a great job. We are in lab 5, which talks about homeostasis and an introduction to anatomy. Your lab 4 quiz will also be due this Sunday, as well as this worksheet. I haven't made your lab 4 quiz yet, so look for it to be available by tomorrow, and I'll get that to you guys. So lab 5, in the lab 5 module, you guys have the lab guide as well as the worksheet like normal to kind of walk you through the lab. This lab will be a lot more simple than last week's lab. I think you've talked about homeostasis in your lecture. This is the process by which your bodies maintain a constant internal environment. It uses your hypothalamus, a part of the brain, which is located just right above your brain stem. And it forms a group of neurons that forms a link between your nervous system and your endocrine system to help to maintain normal bodily function so that everything is constant. That's homeostasis. Most of your body systems maintain homeostasis by what we call negative feedback, which is just the way your body responds to a change to try to get it back to normal. This occurs when a stimulus or any sort of environmental change, this can be a change in temperature, for example, is met by a response that will reverse or negate the trend of that stimulus. So that's why we call it negative feedback. And as a result, your internal environment is returned to normal. There are many body systems involved in homeostasis, and we'll investigate some of these in this lab today. And an example that you will see firsthand, specifically in today's lab, is about the maintenance of body temperature. There's many parts of the body that help to maintain your body temperature, and they help to maintain your body temperature around what we call a set point or a fixed regulation point. And in our bodies, normal constant body temperature is 37 degrees Celsius or 98.7 degrees Fahrenheit. And if you have an increase or a decrease in change in temperature, the hypothalamus receptor will detect that change in temperature and try to produce some effector changes in your body to revert the body's temperature change back to normal or back to this fixed regular point. So for example, if this fixed regulation point is 37 degrees Celsius, our temperature, if we have any increase in body temperature, that can be due to exercise, being in the sun. The hypothalamus detects that change and then sends out a response so that we'll begin to sweat. Our hairs will lie flat and our blood vessels will vasodilate to try to decrease some of the temperature or dissipate that heat to bring our body temperature back to normal. If we have a fall in temperature and temperature decreases below the set value, your hypothalamus will detect that change and then send out a response to the effectors to vasoconstrict your blood vessels. So your blood vessels will constrict to try to maintain your blood and body temperature near your core organs. It'll also create goosebumps and your hair will rise. Your skeletal muscles might start to shiver to try to increase heat to get us back up to normal. So that's this idea of negative feedback and how your body maintains these negative feedback mechanisms to keep all of your body's processes around a normal set point. So you will look at the effect of exercise on body temperature. And I think for this one, I think first you'll be doing some of these by yourself. You want to write a testable statement as a hypothesis, indicating your expectations for the experimental outcomes. So for example, you guys will be engaging in some periods of exercise and rest. And an example of a hypothesis that you are going to write will be increases in body temperature due to exercise will initiate negative feedback mechanisms like sweating that will return the body temperature to normal following a rest period. So that's an example of a hypothesis. Try to think of a new one. What you're going to be doing is you're going to be taking body temperature or looking at the lab guide to see what the body temperature is. And basically what you want to talk about is, will exercise increase or decrease your body temperature? And your hypothesis can be as simple as that. Exercise will decrease your body temperature, for example. So let's take a look at what your lab guide says. Take a look at the safety precautions and the procedures, but our lab guide will take us through a little bit more what you'll need to do. So here's the lab guide. Again, this will be much more simple. The lab instructions, first of all, will indicate that this lab activity is done in groups, but you can do it alone, but only do so if you're in the good physical condition to perform 2 minutes of vigorous exercise for 3 separate trials. If you yourself can't do that, can you get your kid to do that for you or a significant other? And if you have a thermometer of some kind. And the person who created this lab did this lab at home to collect data, just in case any of you needed it. So if you don't have a thermometer at home and you don't want to do the exercises yourself, I think she does include the data for her own trials of exercise. But if you want to do it yourself to see how your own body responds to exercise and the change in temperature, you're free to do that. This takes you through how to write that hypothesis statement, and it should always be an if-then statement. So what that means is, if I exercise, then my body temperature will decrease. That's probably not right. You probably want to say, if I exercise, then my body temperature will increase. So make sure your hypothesis is in an if-then statement. Read the safety precautions, ask someone in your household who is healthy to be your experimental subject, or you can be the experimental subject yourself, or you can use the data that they provided. All right, so you will enter the data for this experiment in the tables on pages 3 and 4. Data, and this is the data for those who need it. And please, if you can obtain your own data, try to, because it's just more fun if you're obtaining your own data. So I'm going to go back to the worksheet and see what we're doing here. So the procedure is, you're going to start by determining some person to be the experimental person, and you're going to record your temperature, your body temperature first in Celsius if you can, in table 1 at rest. So you'll record the temperature at rest first of all, and then your subject will do 2 minutes of exercise. This can be marching, jogging in place, jumping jacks. It can be walking upstairs for 2 minutes. Then you'll record the temperature on the forehead, or if you have an oral thermometer, after 2 minutes of activity, and you'll record that temperature down. So what you're doing here is what you should do it with an experimental subject and with a control subject. So you can do it, by yourself, and you can be the experimental subject. The control subject can just be resting temperature for both. And then, so you'll record the temperature on both at rest, and then you'll record temperature after 2 minutes of vigorous exercise and then after 5 minutes of rest. So you'll exercise for 2 minutes, record your temperature, and then you'll rest for 5 minutes, and then you'll record your temperature as well. And I think you just do it again for another trial. So you'll exercise again for 2 minutes, record the temperature, and then after 5 minutes of rest, you'll record the temperature as well. If you have a control subject, another person in your home who's just sitting around doing nothing, you can control their temperature. Most likely the one who's doing the exercise will have more. But again, if you need data or do not want to go through this, there is sets of data in the lab guide that you can take. And you're going to do it 3 times actually. So this is the 3rd trial. And then what you're going to do is create a bar graph for body temperature recorded in these first 4 tables with a graph. And you'll need to label the x-axis the trial number, enable the y-axis the temperature. You'll provide an appropriate title for your graph. When in doubt, your title of your graph can just be temperature versus trial number of exercises. Your resting data will be displayed as 2 columns, one for the experimental subject and one for the control. And then your trials 1, 2, 3 will be displayed as 3 columns each, one for the experimental subject after exercise, the experimental subject after rest, and one for the control, which will always hopefully be just 37 degrees. So what I mean by this, so you'll want a title on the graph, which you can put as, trial number versus body temperature. On the x-axis, you're going to be putting, or the y-axis, which is where my cursor is, the one that's going up and down, will be body temperature. And on the x-axis will be the trial number. So the first kind of set of columns, and they take you through this, will just be resting data. So you'll have 2 columns here, 1 column, 2 columns. There'll be the resting data. 1 column will be for the control, will be for the experimental subject. And then you'll have 3 sets of 3 columns for each trial. So you'll kind of have 3 groups of 3, and in each trial, you'll have the experimental versus the control after 2 minutes and after resting, and then after 5 minutes of resting. Then you'll write a conclusion asking if your hypothesis was correct or not. And that should be probably true. You should probably see an increase in temperature as you've been exercising, and then a decrease in temperature as your body rests again. So here's the data continued. You can again use this data that they give you for each trial. This is trial 2 and then trial 3, giving the 2 minutes of vigorous activity and then after 5 minutes of rest. And then you'll use this to generate a graph. And again, they take you through the instructions on how to generate that graph again. If you have questions about how to generate that graph, let me know. And then part 2 of this activity takes you through body cavities and body sections. You can use your human biology book for a little bit more introductory material. Let's take us back through the worksheet. So this is the introduction to anatomy, taking us through the body cavities and body sections. You'll need to read this section on body cavities and then labeling these numbers of body cavities using your textbook or another resource to help you label the different body cavities that are shown here. Number 2 refers to the dorsal cavity, 5 is the ventral cavity, and then within you have different cavities listed. I know you go over this in lecture, or if you Google body cavities, you'll be able to find a lot of resources that will help you label pictures like this. Some that you'll need cranial cavity, spinal cavity, abdominal, thoracic, pelvic, abdominal pelvic cavities. So you should be able to do that with the resources that you have labeling these cavities. Then you'll be reviewing some of the directional anatomical terms and body planes: anterior, posterior, superior, inferior, lateral, medial. So all of these directional terms have to do with referring to body structures in anatomical position. An anatomical position is shown here: someone standing up straight with their palms facing forward. Prone describes body that's lying down in a face down orientation. So think about like getting a massage, prone. And then supine describes the body that's lying down in a face up orientation. So here are kind of descriptions of some of these directional terms and how you will describe them. And you're going to use these directional terms in anatomical position to do a dissection. You'll also just be going through reviewing the planes of the body. The transverse plane of the body separates the body into a superior and an inferior part. The frontal or coronal plane separates the body into an anterior and a posterior part. And a sagittal plane separates the body into right and left halves. And understanding these body planes and the cuts that they make, so a sagittal cut would be down the midline, a transverse cut would be separated into a top and bottom, and a frontal cut would separate into a front and back cut. You guys will kind of just use some directional terminology to describe the following structures. So I'll go through a couple of these with you. So for example, the neck is what to the eyes, and this would be medial. So the neck is medial to the eyes because the eyes are more lateral and the neck is medial. A couple of the directional terms could work for this. The neck is also inferior to the eyes. So you might have more than one option. So you'll need to use directional terms to describe these structures to each other. The elbow to the wrist, so your elbow to your wrist, the elbow is proximal to your wrist because the elbow is closer to the point of attachment or closer to your shoulder. The elbow in anatomical position is also superior to the wrist. So remember these, you're describing, oh, did not mean to do that. You're describing structures in relationship to each other, and you'll be describing them using these directional terms: medial, lateral, inferior, superior, and going through. Have you guys gone through this in lecture? If you could just write in the chat yes or no, that might help me. And if you guys have questions on any of those, let me know. A lot of you are working with a lab group, which is great. So you can work with your lab group to fill those out. But if you have questions, send me a message and I can talk you through that, or come see me in my office hours tomorrow. Again, if you are working in a lab group, just be sure to submit yours individually. Sometimes I'll give you a 0, but then just let me know later if you have submitted that. The third activity, and before we get to the third activity, let's see if your lab guide has any more information. Not really, because then it takes you through to your dissection. So you will be doing an in-home dissection. You'll be doing an in-home dissection with a banana, and you can use a cutting board, a baking sheet, plate, or another item. And you're just going to use a kitchen knife instead of a scalpel. You can use forceps or tweezers, but you don't really need pins. If you don't have a banana at home, you can do an appropriate food item to use as a substitute. And this is just as a YouTube video that helps you insert images onto your Word document sheets. But let me go through and talk about how we're going to dissect the banana. I'm just reading activity 2. For each problem, we only use each term once. Yeah, so for activity 2, good question, Yvonne. There's not necessarily, you won't necessarily use all the terms. You could use a term more than once. Good question. So they give you a bunch of terms, but you might use terms more than once. You might not even use some of those terms. Good question. So using the banana, and I think the banana really helps you to do this dissection. And if you're working in a group, maybe one of you is dissecting the banana and others of you, they can tell you about it. So you're going to describe the superficial layer of your specimen, and that's the banana peel. So you can just say, well, this is the peel. So we're trying to kind of give you an idea of how to dissect a specimen, and think about, if it was the human, the superficial layer would be the skin. In the banana, it'll be yellow, it'll be kind of soft, hard, so that'll be the skin. And then you're going to assume that the dorsal side is convex or bows outward, and the ventral side is concave and bows inward. The stem is the cranial end, and draw a profile view of your specimen in the space provided. And to do that, so you're going to draw a picture here of a profile view of your specimen. And what we're trying to show you here, and I'll try to just kind of describe what I want. We want you to draw a side view of the banana, and you can even probably do yellow. So it looks like something like this, and with the stem is at the top. So this will be the superior or the head view of the banana. And then it's saying that the dorsal side is the posterior view, and the ventral side is the anterior view, and the dorsal side will bow outward. So what we're just trying to show you here is that this will be the dorsal side or the posterior side of the banana. And mine is not the best. Here's the posterior side, and the anterior side will be this part. And we're looking at the banana from the side. And then this will be kind of the head or the caudal end. And I'm not going to even undo that. So that'll be the posterior and the anterior view. So you'll want to draw that down there. The stem is the cranial end, excuse me. So that's kind of where idea of our specimen is. So here's kind of the head. I'm just going to write that here. So here we have the cranial end, or the head is by the stem, and then you have an anterior and a posterior view. So you guys will draw a banana much like this for step 1. So that's illustration 1, just to get you oriented that that's your anterior view and your posterior side of the banana, and the head is there. So that's kind of your first step or first drawing. Measure the length of your specimen from cranial to caudal end. So from the stem going all the way down to the end, you'll measure it in centimeters. So what you'll do is you'll take a ruler and measure from the top of the stem all the way down to the bottom of the tip of the banana. And you'll measure that in centimeters to get your centimeter measurement. You'll label both sides in your illustration. And then you will hold your banana so that you're looking at the anterior surface. So you're going to turn your banana around so you're looking at the anterior or curved inward side. And they want you to draw a circle about halfway down to represent the belly button, because that will help you. So you're going to turn your banana around so you're looking at the anterior side, and you will draw or just kind of use a marker to label a belly button right there, because you're going to look at it from the anterior side. Once you've labeled the belly button, you can label a face too if you want, and you'll start to dissect the banana, and it will take you through those steps here. Beginning at the cranial end on the anterior surface, make a superficial mid-sagittal cut to the caudal end. So you're going to go on the anterior side, right down the belly button from the top of the banana to the end. You're making a superficial cut. So that means you're just cutting through the peel of the banana. Do not cut into the deep tissue. So don't cut through the banana fruit itself. At the location of the anterior surface where you marked the navel, make another superficial cut, this time along the transverse plane. So remember, the transverse plane separates out specimens into a top and bottom half. So what you're going to do is you're going to cut through the skin of the banana. Let's say I'm the banana, cut down through to the belly button. When you get to the belly button, you're going to cut along the sides through the transverse plane. So what you're doing is you're going to kind of cut or peel back the peel. Again, not cutting into the deep tissue yet. So you're just cutting through the superficial or the peel. Now you should be able to peel back the integumentary covering. You can place pins at an angle. Don't worry about pins, just peel off the parts of the banana. Measure halfway down the length of your specimen. Beginning at this midpoint, make a complete transverse cut that it does extend through the deep tissue. So halfway through your specimen should be about at the belly button region that you labeled, and you're going to make a complete transverse cut. So what that means, if my water bottle is the banana, a transverse cut separates specimens into a top and bottom. So you will completely cut through your bottom or your banana halfway to create a top and a bottom portion. Then you'll start at the superior end, make a deep mid-sagittal cut to the caudal blend. This was I think some of this takes you through pretty well, these steps. Let's see. You'll make a coronal cut through the entire specimen. And remember, a coronal cut is cutting through a specimen, separating a front to the back. So if we go down to your first picture of the banana, a coronal cut would be down the side to separate out the anterior from the posterior. Draw the results of your dissection when finished. Call your instructor to your station. So on illustration 2, you're going to just draw out the pieces of the banana that you've cut out. Dispose of your specimen in the trash. So it's a simple dissection, but it's to help you practice using the body planes, the coronal section, the transverse section. So it seemed like a simple activity, but it's really helpful to kind of get into your head some of these different activities. So that's for this week's lab. I'll go ahead and stop the recording and answer some questions for those of you guys who are still here.